Imagine that you have an identical twin who lived in a different place where she learned to do different things. For example, your twin smoked many cigarettes every day and she loved eating junk food. She even gained much more weight than you. With time, you will notice that both of you look different and behave differently. Your twin might even have a shorter life than you are, as she might develop diseases that you don't have. But how come this is possible? And you are both identical twins. Identical twins have identical DNA. This means that you both have the same genes with the same sequence in your DNA. But how come you both look different, you behave differently, and one had diseases while the other did not? This is because you have different factors controlling your genes. Some of your genes may be switched off in your DNA, but maybe something caused them to switch on in your twin's DNA. This is all because of epigenetics. Epigenetics control genes. Epigenetics literally means above genes. It is what switches genes on and off. Where you live, what you do, what you eat, your work, how you exercise. All of these can affect the way genes are expressed in your body over time and cause some genes to switch on while others to switch off. Epigenetics makes us unique. What controls our genes makes each one of us unique. Why some of us are more active than others. Why some like milk while others do not. Why some develop diseases that we do not have. All of this is because of gene expression. Gene expression differs from one person to another. So what could regulate transcription and gene expression in us? There are several factors that may control the expression of your genes, that may switch them on and others off. First, there are proteins that bind to specific non-coding regions in your DNA. These proteins can regulate how much transcription of a particular gene will take place. There are some proteins that may bind to the enhancers, which are non-coding regions that increase the rate of transcription when proteins bind to them. Other proteins may decrease the rate of transcription when they bind to silencers, which are non-coding regions that lower transcription when proteins bind to them. We also mentioned before that there are proteins that initiate transcription when they bind to promoters on the DNA or the promoter proximal elements on the DNA. In addition to the regulatory proteins that bind to non-coding regions of DNA, the supercoiling of nucleosomes in your chromosomes helps to regulate transcription. Remember, we said that within your chromosomes, the DNA is wrapped around 8 histones to make up a nucleosome. Look at this picture of nucleosomes. Note that there are areas where the DNA is tightly packed and the gene is not exposed, while in other areas the gene is exposed and accessible for the production of mRNA by transcription. Two things may affect nucleosome supercoiling and packing up. When acetyl groups are added to the positively charged histone tails, they become negative and the DNA repels against them, causing the nucleosome to open up and allowing the gene to be transcribed more often. If histone acetylation does not happen, the DNA remains tightly packed and transcription is inhibited. In contrast, nucleosomes tightly pack when a methyl group is added to the histone proteins. Methylation of the histone proteins inhibits the area of the DNA to be accessible for transcription. This decreases transcription of the gene. The amount of methylation can change over time in each individual and can be affected by environmental factors. Note that until now, we have mentioned two functions for nucleosomes: supercoiling of chromosomes and regulating transcription and gene expression. Environmental factors may also have an impact on gene expression. Temperature and light could affect how genes are expressed in some organisms. Let me give you an example. Himalayan rabbits appear as completely white in summer when the temperature is higher than 35 degrees Celsius. while in winter, note the color of the rabbit's ears, tip of the nose, and feet. When the temperature is much lower than 35 degrees Celsius, some parts in the rabbit turn black. This is because of a gene in the Himalayan rabbit called the C gene, which causes the formation of black pigments in some parts of the rabbit's body. Temperature affects the expression of the C gene. The C gene is inactive above 35 degrees Celsius. In warm temperature, the gene is inactive and no pigments are produced. Therefore, the fur color is white. And it is maximally active from 15 degrees Celsius to 25 degrees Celsius. In winter, the C gene is active and pigments are produced in some parts of the rabbit's body. Another example in humans. UV light activates the production of melanin in human skin, causing skin color to become darker. Regulatory proteins, acetylation or methylation of histones, and environmental factors all affect DNA transcription and how genes are expressed in our body. If we could identify all the factors that cause the different genes in our body to switch on and off, we would be able to reverse the gene state to keep the good ones switched on and the bad ones switched off. Then probably we could cure diseases like diabetes and cancer, slow aging, stop obesity, and so much more. See you soon.